Today we are going to discuss the basic nature of MPC and then move on to two conditions that can result from a deactivation of this complex. Lastly, we will discuss some possible remedies when deactivation occurs. In order to understand what exactly MPC is, we have to look into the cytosol and mitochondria and then take a look back at previous metabolic pathways that we have discussed in class. Looking back at glycolysis, we can remember that pyruvate is created from glucose and also that 2 net ATP are generated. So this is very important because pyruvate can have a wide variety of fates once it is produced. So it can either go into the mitochondria for further modification via oxidase, or it can be produced into lactate, which can then be retaken up by gluconeogenesis and produced back into glucose if the organism is not receiving enough glucose through its diet. As we know, there are many different fates of pyruvate. If the organism needs more energy, then it will probably enter through the mitochondria. For instance, once this happens, it will be oxidized to acetyl-CoA by pyruvate dehydrogenase and will then enter the TCA cycle. This produces around 32 net ATP. Or it can be carboxylated to oxaloacetate by pyruvate carboxylase, and this will either enter the TCA cycle once again, or it will be taken out of the cytosol and be used for gluconeogenesis. Now, what happens if it does not enter the mitochondria at all? Well, pyruvate will just stay within the cytosol, and then it will be reduced to lactate via lactate dehydrogenase. Although this produces less ATP than oxphos, it will still produce some NAD plus for glycolysis. So that way, once gluconeogenesis has occurred, the NAD plus will still be available for more pyruvate to be made. I have said the term MPC a lot, and you might be wondering, what exactly is that? MPC stands for the mitochondrial pyruvate carrier, and as the name implies, pyruvate, which is currently in the cytosol, will be brought through this complex into the mitochondria, and once it is here, it will be either oxidized or reduced, and then it will be able to enter many different cycles that we see here. This is very important to cell sustainability and development, and that is why the MPC's function of essentially being the front door to the mitochondria is so essential to cell life. In order to understand how MPC downregulation can lead to many different diseases, we need to understand the structure and location of this. So MPC is a multimeric protein complex, meaning that it consists of 2 or more polypeptide chains, which are the subunits of it. So here you can see the different subunits of MPC. So the location, it is obviously going to be integrated within the inner mitochondrial membrane, which allows for it to connect to the outside, like the cytosol, and bring it into the matrix. So in humans specifically, we have the MPC1 and MPC2. And so each of these are going to contain 2 transmembrane alpha helices. Now that we have gone over the basics of MPC, I would like you to answer the following question. Referring back to previous slides, what is the structure, location, and function of MPC? Now that we have an understanding of MPC, I would like to look at how MPC plays a role in pathological cardiac hypertrophy. Now, what is that exactly? Essentially, this disease is associated with increased cardiomyocytes death, which would be like the cell muscles within the heart, they're dying, and then fibrotic remodeling. So there's not as many cells anymore, so that means that the heart itself would have to remodel so it could be as functional as possible. Here we have 2 different types of hearts. So a healthy heart is going to have 80% of its ATP coming from fatty acid oxidation, and then 20% is going to have its ATP coming from glycolysis, so this gives it around 32 ATP. And as you can see, there's no remodeling, it's just a healthy heart with plenty of movement throughout its arteries and ventricles. And so then we can look at the heart that is failing. And so this heart is going to rely a lot on glycolysis for ATP. And as we know, glycolysis really only has about 2 net ATP, so it is going to be energy deficient. And this means that there is a decrease in the usage of MPC because that pyruvate is not going into the mitochondria, is just staying outside. And so pyruvate is going to be reduced into lactate within the cytosol, making it inefficient in terms of ATP production, and this will contribute to the energy deprived state. According to the Warburg effect, which we talked about in class, cancer cells tend to transform glucose into lactate, and this is very similar to how the heart does once it becomes unhealthy. So both heart muscle cells and cancer cells exhibit limited mitochondrial oxidation, meaning that they are not using their MPC. In order for more to be known about how to cure this issue, they needed to conduct some research. So mice were used and they were basically induced into hypertrophy. So that way we could see the effects of how mice reacted and compare that to human hearts. So once they were induced, it was seen that there was evidence to support the reduction of the carrier abundance can cause pathological myocardial growth. On the last slide, you could see in the failing heart that there was this kind of white stuff that was growing within the heart, and that is essentially what this is talking about. It's bad cells because it's not being transcribed correctly and things like that. And this was also supported by knocking out specific subunits, so just completely taking them out. And this once again showed downregulation, leading to more lactate production. And you could see that this was happening instead of pyruvate entering the mitochondria. And the subunits of the MPC will stabilize each other once they notice that something is wrong and that they're failing, but this does not occur at a transcriptional level. Instead, it happens at the protein level. Additionally, there were TCA cycle intermediates being accumulated downstream. So it is very questionable because how is this occurring if there is no pyruvate entering the mitochondria? How is the TCA cycle still happening? Well, actually what happens is animals develop adaptive routes to supply carbon to the cycle, so that way the energy is still being supplied to the heart. So they're figuring out a way to adapt themselves so the heart can still survive. It is just not doing it at a healthy amount. And this can be mediated by ketone bodies or beta oxidation of fatty acids that are actually replenishing acetyl-CoA so that the cardiac remodeling does not occur. So one way to do this to kind of prevent heart disease was to eat a dietary ketone diet, which would include things like fish and oils, and it was proven to be very efficient in preventing heart disease in the mice who had their MPCs knocked out. And this is because the ketones are able to change myocardial structure and function and even correct metabolic dysfunction that might occur due to the decreased MPC expression. And although that is happen on most cases, the mice and humans would still develop the pathological hypertrophy simply because you can only do so much. You still need that MPC to be functional in order for you to have a truly functional heart. There was a need for a reason behind this disease. So many thought at the beginning that it was just metabolic remodeling. So essentially meaning that different processes were going to be used, and that is why. But that is not why. ATP is still going to be generated. The body is still going to figure out a way to survive. But the issue comes when the person is older and they have generated so much damage to their body without even knowing it. Because ultimately, the heart is still getting its energy, but the wrong pathways are being used, because there are a lot of different pathways that kind of do the same thing and have the same products, so that way there's a redundancy if one fails. But when you get older, you can have so many different health problems that even if one fails, there might not be anything you can do. So, another thing that was very, very promising was post-translational um, protein modifications. So, a lot of acylation was occurring in the proteins of MPC1, and this was occurring due to glucose going to the hexoacidamine pathways and not going to the pentose phosphate pathway. And it is not going there to that pathway when MPC has been deleted in adulthood, because glucose is once again being broken down somewhere that it should not be. So these modifications have been stated as a possible cause of heart disease and failure. And although that is true, the mechanisms of MPC downregulation are still being explored, and I will cover a few more of these later on. We have covered everything about hypertrophy, so now I want you to answer one last question. How is the TCA cycle still occurring if pyruvate is not entering the mitochondria via MPC? Next, we are going to look at ischemia reperfusion injury and how MPC plays a role in this. What is IRJ? Essentially, this is an inflammatory response to the MPC activity within the heart changing, and the pH is going to also change. And so, as you can see here, some bubbles or just pockets of material is going to form within the heart tissue, and that is going to cause some difficulties with blood flow. And so, what is the importance of this? So the pH, as we all know, is a major modulator within any biological system. And within the heart, it is extremely important for how much activity is going on. So in a normal system, the metabolic products will just flow straight through the MPC to the mitochondria and then go through metabolic cycles. If the pyruvate is not transported efficiently, like when MPC is inhibited, it will be reduced to lactate, and this can cause acidosis, which means that essentially the intracellular pH can decrease to a very acidic level, which can make the extracellular environment then do the same, and the whole environment just becomes so acidic that nothing can thrive there. And then there will also be some swelling due to osmosis, which you can see here in this picture. This is just so much swelling and it can't control it anymore. And then there's also an increased MPC in those who survive this disease. So if they started off with a low MPC activity rate, but then they saw an increase near the infarct zones, they will be able to survive most likely, because their body is going to kind of go back in time and try to make up for its lost cause. Like I stated earlier, the acylation of lysines is the most likely culprit as to why the MPC is being blocked, as you can see in this picture. But if you deacylate these lysines via SIRT3, you can then open back up this MPC and pyruvate will be able to enter, leading to metabolic alterations. So this can happen via medication, and this will really help the individuals that suffer from these diseases. Lastly, these are the resources that I used, and thank you guys so much for listening.